Welcome to our discussion on a groundbreaking discovery in neuroscience, finding neural stem cells outside the brain and spinal cord.
Wow, that's quite a twist on what we've always believed, right?
Absolutely. Scientists have now identified these peripheral neural stem cells, or PNSCs, in tissues like the lung and tail of mice.
Yep, that's wild.
It really overturns decades of neuroscience dogma.
So these cells, they behave just like the ones we find in the brain?
Exactly, they share the same key properties: self-renewal, differentiation into neurons, similar gene expression profiles, and all that stuff.
Interesting.
And unlike neural crest stem cells, these PNSCs can sustain neurogenesis for a longer period.
That's pretty impressive. And it means we could potentially use them for therapies, right?
Right on. It opens up a whole new avenue for treating conditions like Parkinson's or even repairing spinal cord injuries. I mean, if we can find these cells in humans, they could be a game changer.
This does sound revolutionary. It almost feels like stumbling on a hidden treasure trove in what we thought was a well-mapped territory.
Exactly. It challenges the long-held belief that neural stem cells are confined to the CNS. Imagine, we've got these cells residing in more accessible tissues. It's like discovering a secret backdoor to neural regeneration.
Yeah, really neat. And when it comes to cellular therapy, having an easily accessible source is huge. But then, how reliable are these cells in terms of self-renewal and differentiation?
The studies show that PNSCs mirror brain-derived NSCs almost perfectly, including how they maintain their properties over time. They even express NSC-specific markers and have similar epigenetic profiles.
It's also fascinating that this discovery came about through efforts to replicate earlier, controversial findings, right?
Yep, the whole STAP method fiasco. Their initial goal was to reproduce pluripotency via low pH treatment, but instead, they uncovered these PNSCs. It's amazing how sometimes even failed experiments can lead to major breakthroughs.
I see the potential there, but don't you think we should be cautious? Could these peripheral cells behave differently in the human body than in mice?
That's a valid concern and definitely something to keep in mind. While data from mice are super promising, translating those findings to humans will require extensive research.
Still, the prospect of non-invasively accessing neural stem cells is pretty enticing for regenerative medicine.
Absolutely. It could reshape how we approach treatment for neurodegenerative diseases. The ease of obtaining these cells compared to brain NSCs lowers a lot of the barriers we currently face.
And that's a huge leap forward over traditional methods of dealing with neural damage. It's like moving from a back-breaking surgery to a gentle, more efficient procedure.
Exactly. And the interdisciplinary collaboration in this study, labs from Europe, Asia, and North America coming together, really reinforces the credibility of these findings.
That's impressive. All in all, this discovery not only deepens our understanding of neural plasticity, but also lights a path toward innovative therapies. I'm excited to see where this leads next.